Okay, everybody, welcome to the call tonight. I appreciate you all being on. You know, you could be doing anything tonight, but you chose to be here with us to learn about Zenoa and new products and how you can utilize that product in your business and expanding your network and also your customer base. And so I have the pleasure tonight of introducing our first speaker. He's the Chief Marketing Officer at Zenoa. And he's the guy who, he and our other speaker are kind of responsible for bringing you these great products. And they have a product path all planned out for this company and for this business, for your business, that'll blow your socks off. And so to get us started in talking about our new products, I want to introduce Chief Marketing Officer David Casteller. David, you're there?
I am here. Thank you, Brad. Appreciate it. It's really great to be here with everyone. Let me just start out. Okay, we there? Sorry, I was getting some feedback.
You're there. Go ahead, buddy.
Okay, well, let me just start by saying I'm really, really excited about this SRQ product. I was one of the test subjects on this product a while ago, and it was such a good product. I actually ran out, and then a week after trying it, it was just unbelievable to me how much I was able to notice the difference from that product when I didn't have it. At first, I noticed that, hey, look, I had more energy, and I felt more awake and alert, kind of more focused. But then when I didn't have it, I really noticed, I said, oh, man, I've really become accustomed to this product. And I was so excited about this. The research that was done on this product is really amazing. I was just in Japan, and the Institute of Applied Biochemistry out of Japan is the the organization that originally developed this product. And we will have some recordings coming out with Dr. Sugiyama, who is the one who initially developed this product. And his whole thing, the Institute of Applied Biochemistry, is really the forefront of anti-aging. And so they were really studying the brain and how do we stop the brain from aging? How do we kind of reverse the oxidation effects that happens to the brain, and particularly the mitochondria? Now, those of you who may not know, and Alpha will probably go into this in more detail, I had to learn a lot of this just like you. But the mitochondria, they're your energy center of your cell. And this product was really developed initially to fight oxidation and improve the mitochondria in the cell so the mitochondria are more efficient. And while being in Japan last week, it was so interesting to learn more about this product and how it actually works, some of the science behind it. And Alpha will get into that. And I'm so excited to have Alpha on board here because he is such a knowledge base to help us, and he's so good at being able to explain in easy to understand terms different products and reasons for the science behind different products. Let me just say also that this SRQ product, I believe the shipping date will be the first week of January. And Brad, if you want to correct me on that, I just want people to understand that that's the date that I understand it will be shipping out here in the USA. In Japan, I think it's January 15th.
That's right.
So we have some great products coming out, and I'm so excited about them. We've had some affiliates that have called and asked for different marketing materials and given us some ideas and things like that. And I want you to know that we'd love you to send those recommendations and thoughts that you have. We are brand new and we are in pre-launch, but we are taking those and we're trying really hard to be mindful of what you need and what tools you need here at Zenoa. As we said before, and you've heard this before, Zenoa is about freedom and about really trying to become a company that's different. And the way we want to be different, well, one of the ways is by listening to and really working with our affiliates for product development and for materials. So please let us know as you think of and come up with ideas for marketing or anything like that. We're very excited to have you, and thanks for being with us in such a early stage here. Well, Brad, is it okay if I introduce Afa real quick?
That would be awesome. Thank you.
So Afa, I've known Afa actually for about, I want to say 13, 14 years now. Afa is a PhD and is amazing, like I said before, at being able to explain science in an easy, kind of a normal person speak, I should say. He's just able to explain it to people in a way that makes sense. One of the things that I love about working with Afa now is in marketing, I'm trying to figure out, and everyone always is, well, how do I explain this to someone? What does this ingredient do? And so Afa has been the one to help us organize research, the medical studies that actually shows, hey, Phyllosea reticulata or quercetin or resveratrol, this is what it does. This is why we want it in this product, in this formulation. This is the research that's been done on it. So, Afa, let me turn the time over to you, and I'm sure everyone's excited to hear you. We've got tons of people on this call, and I'm really excited to hear what you have to say. And let me just tell everyone one last thing. After being in Japan and out talking to Brad Reese, and many of you have talked to him and some of you have met him, there are literally tens of thousands of people waiting to get on board at Zenoa. And I just think the opportunity is amazing. I think you guys are in a perfect position. I'm excited as ever for 2017. We're going to wrap it up strong. NOAA shares do end at the end of the month. So anyone that's maybe interested, and if you're interested in getting those shares up, then now's the time to really be pushing people. The end of the year is when it ends. So we did that in part just to say thank you and help everyone get a part of the company as we go forward and really grow in the growth. I mean, be able to participate in the growth of the company as we move forward. So, Afa, let's turn the time over to you. We're excited to hear what you have to say.
Thank you, Dave. Appreciate the introduction and the opportunity to connect with our affiliates that are out there. I want you, the affiliates, to know I have so much respect for you because of what you do out there. That is the hardest part of our business. But I also want you to know that we love you and we want to do the things right all the time. That is the direction that John Wadsworth has given us. So let me go through the slides and explain to you. I'll try to stay as simple as I could. And if you have any questions, I would not mind if you send me the questions to my email at afa@zenoa.com. So if you have a question that is just dying to hear the answer, send me an email and we'll have a conversation via the email. So this product, SRQ, is one of my favorites. I know David said that he's excited about the product. I would say I'm much more excited than David, if I may say that. So, and I will give you only a few, but please note that the benefits of this product far exceeds what we have that I will share with you tonight. I just wanted to point out a few things: the anti-aging effect of this product, the antioxidant effect, the natural energy that you get from taking this product. It does not have any stimulant of any kind. And also the management of blood sugar that we have. That is perhaps one of the biggest, I would say, advantage to this product. But it cannot be, we cannot live in a place where we're all locked up in a room and then we say, well, this product is only good for this blood sugar management. No, it's much more than that. So when we do any formulations of any kind or make any products of any kind, there are three tenets that I want you to know that we go through in order to put together the products and the ingredients that comes with it. Number one, the ingredients that we are going to choose, it has to be of the highest quality. We cannot have any synthetics of any kind in it. It has to be natural. That's number one. Number two, these ingredients have been shown to work in humans, not just in the cells, not just in mice or rats or any other animals that have been used for research before, but it shows that it works in humans, because we are all humans after all. And that is for a good reason, because there are sometimes research that are done on mice or rats, even though they have similarity towards our immune system and things like that, sometimes they work in mice but not in humans. And the last but not the least, they're safe
So,
for humans.
Alpha, can I just ask one question on that?
Sure.
Is that common for companies to release products that haven't really been proven effective on humans? Is that common? Is that something that normally happens?
Yeah, it's very common. A lot of people probably don't know that, but I like to say these are ingredients that have been shown to work, and we put the ingredients, of course, in a different dynamic, and it always works. I mean, we have SRQ has already been out, and a lot of people have sent in some questions and sent in their testimonials, and we know this ingredient works. So that's why it's important to know not just the quality, not just it works in humans, but also it's safe for human consumptions. I'm not sure if I did answer your question directly, David, or...
That's perfect. Yep. Continue. Sorry.
Okay. So that is the last part, they're safe for human consumption. So all of those three major parts that guide us in how to come up or make a product for a specific reason. If they don't meet all of them, then it does not make any sense to make a product. So it's very important, and I wanted to share with you so that you know what we work through. Ingredients got to be the highest quality. It works in humans, and it's safe for human consumption. So I would like to watch a little movie here, if we could. Thank you, David. Appreciate you guys putting together that video. I wanted to use that video because it gives an overview of SRQ products. And then I'd like to get into a little bit more details
Rafa, can I just interrupt for 1 second? The sound was very low, so I know our audience didn't really hear it. I got to see it, and it's a great video. And David, if you're still there, maybe I can get permission to commit that we could have that up on the website tomorrow so people could view that, that video and be able to start to really understand this product. David, are you there?
Yes, and that can be. It's actually on YouTube as well, and I'll make sure it gets posted tonight.
Okay, fantastic. Thank you. I'm sorry to interrupt, Rafa. Go ahead.
Oh, no, that's okay. So this product optimization is always start from the ingredients. As I said before, we gotta have the highest quality ingredients. It's gotta be safe, and it has been proven to work in humans. So Salacia reticulata is a well-known plant that has majority of the people are familiar with it from Sri Lanka, but also in India and other places. And we have supporting ingredients like the L-arginine, the L-asparagine, CoQ10, cinnamon, and we have fenugreek seed extract, Momodica carantia, resveratrol, the alpha lipoic acid, and the last but not the least, quercetin. These are not in any order of importance. So now I'd like to show you a little bit of the research that have been done on this plant and also the major ingredients or the major compounds of this plant, Salacia reticulata. In one of these studies, the Yoshikawa were looking at, and they wanted to see what is going on with this plant, Salacia reticulata. Do they have any antioxidant effects? Does this plant have any protection for the liver? You know, the liver is an important organ of ourselves because it does, among things that the liver does is to protect you. So it takes all of the, ideally take all of the things that we eat and drink and then digest those things out and make sure that we do not have any toxic compounds. And you can see the important function of the liver. So if there are any toxicity materials that get into the liver, does this plant have any effect on the liver itself? So they designed an interesting study, and they were looking at mice. And what they want to do is something that is done regularly in research. They look at tetrachloride, or CCl4, is one of the most toxic compound that is used against the liver. And so they inject this compound into the mice and then see what happened to the mice. And they can see the toxicity, the mice is dying and have a lot of other diseases. And then they inject some of the mice with the liver, the same toxicity, and then they follow it up with some extracts of the Salacia reticulata. And then they also have another group of mice that they inject them with Salacia reticulata extract before, and then they add the toxic material, CCl4, and then they look at other things. One of the things that you have to look at, the enzymes associated with the liver. When you see those enzymes that are being dumped into the blood circulation, that means the liver has been compromised or there's an insulin in the liver. So that's why you have to take a blood sample and then you take a look at it to see if they're really up or they're down or there's nothing going on with them. So I picked two of those enzymes here, not because I don't want to use the other one, but because of time. I make sure that we stay within the hour that I was told to stay within. So two biggest enzymes we're looking at glutamic oxaloacetic transaminase, so GOT, and then you have the glutamic pyruvic and transaminase, or GPT. So you have those two enzymes, and you take the blood samples and check to see what's happened. So, of course, after they inject the mice with these toxic materials, both of these enzymes levels in the blood circulations went way up significantly. So it means that the liver has been compromised, or in other words, there is an injury to the liver because of these toxic compounds that have been used. And that's the reaction that you get from mice. So, and then following up this experiment, they put the Salacia reticulata extract into the mice, and what they saw was the GOT enzymes actually decreases by 45.8%, and concomitantly, the GPT enzymes decreased by 67.6% compared to the control. The control, one of the controls, the positive control, did not have any Salacia other than just the CCl4 toxic compounds. So by comparing those two, you recognize that Salacia reticulata actually protect the liver from these toxic compounds. This is an incredible plant, an incredible compound that we are looking at. So, and they look at the other part of the antioxidant. Does this plant has any antioxidant? This is very important to know because the older we get, the more oxidant that we have. I like to think of the oxidant as one of the best video game, I believe, that first came to the island, the Pac-Man. That is one of the best video games that keep all the kids up until, or especially on weekends, until it's almost daylight. Everybody is fascinated with this Pac-Man. And the oxidants in our body is kind of like those Pac-Man things. The antioxidant are the ones that come in and neutralize or take care of those oxidants. Now, the nasty thing about oxidants, if I may say that, oxidants, they're usually in a form of a negative, and then they attack anything that is positive. So they attack the muscles, they attack eyes, they attack enzymes, they attack anything that has a positive sign in it. And this is why it's important that you have an antioxidant in order to neutralize that or bring the amount of the oxidant level down. Now, there is no such thing as you take an antioxidant and it actually zero out the oxidants in the human body. That does not happen in this world. What it does, there is a level of oxidants that are needed in the body. And for a very good reason, because oxidants also become antimicrobials. As you can see, you know, it is true that microbes are everywhere, even in the human body, inside and outside. So there's a certain level that needed to be there. And for the sake of argument purposes, let's just say that it has to be 40% to zero, but you'll never get to zero. I guess it gets you zero when you die. So anyway, so let's suppose that it's supposed to sit not more than 40. And when it gets beyond 40% oxidants level, then it becomes counterproductive and become problematic to the human body. And when it gets beyond 60%, then a lot of these things, for example, the mitochondria will start committing suicide, the cell suicide, or what we normally call apoptosis. So this is very important that we make sure we have the antioxidant to help out in order to take care of the excess oxidants that are out there. And there are so many of them, they come in every hour and every day. As we eat, as we drink, as we go exercise, we become stressful. All of these, there are intrinsic factors, meaning factors inside the body, and there are also extrinsic factors, meaning outside the body. All of these stresses out the body increases the oxidant. Therefore, you need a strong antioxidant such as Salacia reticulata. And it shows in here, if you're looking at it, what is the actual concentration of this plant. The first one is from the root, that is your water extract. And the second is the methanol extract. You recognize that they're very, very active, meaning there is about 5.1 microgram per milliliter that it would take to inhibit 50% of the TPPH. That is the radical scavenging activities. Okay? So the idea here is this: the lesser the amount, the more active that compound is. So, and if you look at the methanol extract, it's about 3.1. So you can say that the methanol extract is more active because it only takes 3.1 microgram per mil to inhibit 50% activity of the TPPH compared to 5.1. Now, and this is why I wanted to show this graph here. And if you look at number 1, 2, 3, 4, 5, 6, okay, all of those are actually compounds from the Salacia reticulata plant. They exist in different concentration. If you go from the roots to the stem, to the bud, the leaves. And I also believe it actually happened in the fruits as well. But I have yet to find any studies looking at the fruit. I'm sure they're out there. But if you look at these major compounds here, let's just look at 1 to 4 and concentrate ourselves on number 1. Okay, menciferin, that is the biggest active compound in the Salacia reticulata plant. It takes about 5.9 micromolar, which is much, 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 much smaller than 5.1 microgram. That shows us that this menciferin compound, which is a part of the Salacia reticulata plant, is very, very active. And not only that, but it's also a strong antioxidant for the human body. So if I wanted to look more closely into it, and you can see there is overlaps of these activities and health benefits that are in the plant. Let's just take the 3 major compounds that are found in this plant. Number 1 that we are familiar with, the menciferin. Number 2, salacinol. And last but not the least, codalenol. Now, there are more compounds than just these 3, but I wanted to focus on these 3 and then just look at 2 of them, because the rest of the compounds, they line up to have the same health benefits which have been shown. Okay, so menciferin, if you look at it, it's useful in management of diabetes, i.e. the blood cholesterol level, and also have been shown that are very strong anti-inflammation. And again, here is one thing that we learned from the slides before: very strong antioxidant, and also very helpful, very, very helpful in the research have been done. They seems to help in the molecular of how things work in a cell, the metabolic and the immunological regulations of the cell, and i.e. the body. So this compound is very active, and it has a lot of activities. Now, the cholesterol-lowering effects of this plant has been shown in animals and also been shown in humans to lower cholesterol and other particles associated with cholesterol. We're talking about the IDL, the VLDL, the LDL, all of those kind of what we call bad cholesterols. And then the last but not least, which I put them on different colors because I want to make emphasis on some of the, it prevents, again, when you have too much of the oxidants in the mitochondria itself, they actually change the morphology, change the shape of the mitochondria, and the mitochondria is no longer effective in producing energy. Okay? So menciferin have been shown to prevent that alterations of the mitochondria due to the excess of oxidants and other materials, and also been very helpful in maintaining the energy production. So we're talking about there is a process in the mitochondria. It's called the electron transport system. This compound actually works and interact with this electron transport system, that it helps to maintain the energy production. That's why you feel energized when you take this plant and this product. And then the last but not the least, if you look at the organ, we go from cell to the organ now. It helps, the research have shown, it helps to maintain the structural integrity of the heart, because heart do change. When you have a lot of the oxygen and other toxic materials in the human body, it stresses the heart and the heart changes. You know, when the heart changes, you don't get much oxygen to breathe, you don't get much blood to go for the rest of the body. And that is one of the reasons why you see this problem in diabetes, among other human conditions. Okay, so if we go from there to salacinol, I want you to know that you see that there are a lot, the compounds itself, they are taken and purified and then used for research. The benefits are basically the same, with little minor differences. If you look at, the first one is the most natural, potent anti-diabetic effects by inhibiting an enzyme. It's called the alpha glucosidase. When you inhibit that enzyme, that means the body cannot produce more out of the food that you eat. So your blood sugar level will stay close to normal. That is very important in order to help us to make sure, especially after, okay, since we're in that area, let me mention this. When you take this product, which I have been taking, it's always better to take this product either together with your food or after you eat. Don't take this product if you haven't eaten anything. Okay? I haven't seen any research that shows that if you wake up in the morning and take this product, it will lower your blood sugar beyond the normal level. However, it is always a good idea to take the product when you're eating or right after you're eating, so that the excess sugars that the body will take the body a little bit longer to digest and bring into the normal range. It actually speed up the whole process. Remember, the older we get, the slower these things becomes. So we need that extra help, that extra push, in order to maintain to fairly normal or reduce to the normal level of blood sugar levels. The second part is suppress the blood glucose. Then the part of this that's exciting, as you can see, the HbA1c. For those who have been to the doctors to check out their blood sugar level, that is part of what we call glycation. That is a glycation end products. And when you have that HbA1c higher, which associate with diabetes or associate with abnormal blood sugar levels, those sugar, they will bind to any proteins or enzymes, and in this case HbA1c bind to the red blood cells. The efficiency of the red blood cells in terms of carrying the oxygen in and then bringing out the carbon dioxide and other waste materials decreases tremendously. This is why it's important. This is why it is part of the anti-aging effects. Not only it lowers the blood sugar level, because the less blood sugar level excess that we have beyond the normal level, the less chances of having elevation in what we call glycation end products, or commonly known as AGEs. Okay? It also improves the glucose tolerance, meaning if you go beyond the normal level, it also helps to bring it down. Okay? And another thing that I found interesting in this research, it actually helped modulate the immune system. What does that mean? Modulation of the immune system simply means that if the immune system is getting more active, it will help bring it down close to normal or where it's supposed to be, because we know that the active immune system are highly active, then you become to have the autoimmune diseases. And interesting, diabetes type 1 is part of those. And some people say that diabetes type 2 is another example, among other diseases. So this is why another good benefits of this plan, this is one of the reasons I'm personally excited about, is the modulation of the immune system. And also shown in the research that it actually increases the natural killer cells. And we should know natural killer cells kills bacteria, viruses, among other things. When they're ineffective, that's when we have a lot of bacterial infection or viral infections. And as the cold temperature coming around, well, I think we know a thing or two about those inability that we have in ourselves in order to kill off those viral and bacterial infections. So that is another good examples and benefits of this plan. Now, to finish off looking at this research was done is another exciting, and I mean, I am so excited about it. And here's the reason why I'm excited about it. So if you look with me, I hope you can see there's part of the graph is on my right side, it's probably on your left side or whichever one, but I have the cursor circling around that graph. And if you look at it, there's something interesting about this, because the anti-glycation activity of this plant, and if you look at, we use the root dose, these guys were working on the root, stem, leaf, and twigs extracts. And if you look at it, it takes a very small amount, very active, to inhibit the process of glycation. Okay. Now, if you look into my left side, where the cursor is right now, there's something interesting about that we're looking at there. They almost look similar, but there's something different about it. And I'll point it out to you in a second here. Okay. Now, this is in the process of making the advanced glycation end products. Okay. So they're in the process of putting the body, how the body puts those together. Now, on this graph here, it shows something very exciting about this plan, okay? Because it shows here in this part of the research using the same materials, the process is already formed, meaning the glycation is already there. Does this plan have the ability to break down those glycation and reverse it? That's what is shown on this graph. So, but the interesting thing, it takes a little bit more. Like for example, the roots is take about 101.6, plus or minus 11.57 microgram per mil to actually break it and reverse it. And if you see on the other side where I have the cursor in, it takes only about 13.06 plus or minus 0.69 microgram per mil. So it takes less from this research. This is an in vitro research. It gives us a lot of knowledge about that it takes less amount of the Salacea reticulata to inhibit the process of anti-glycation. However, reversing those processes that are already there, they already form, it's more like, I like to think of it, it's more like the house is already built here, and now you got to take the house apart and take everything off and leave the lot empty. That's how I'm thinking about it. It might not be the way you're thinking about it, but I apologize. That I have a simple mind, and I like to think of something very simple that will help me to understand. So this has already been established, and then the Salacea reticulata, it takes a little bit more. I'd say comparing those two, take about 10 times a little bit more of the root extract, but it's still very less material. We're talking micrograms now to break out and reverse the process. Okay, so hey Brett, can you hear me? Oh, okay. Now,
yes, I can hear you. Alpha, I can hear you.
Okay, good. So now taking another look at Mensiferin and how it works. Here's a cartoon example of how a mitochondria looks like. And I show it for the purpose of pointing out, here you have your representations of what a mitochondria looks like. If you ever work in a scanning electron microscopy or a transmission electron microscopy, which I was privileged to do a project for 2 years, when you see these things in real life, there is a feeling that comes over you. I'm not sure how to describe that feeling, but here's what they gonna look like in real life. You stain it differently, then they appear differently. And here's what you call the rough endoplasmic reticulum. They're kind of like a pancakes that are put on top of each other. But they appear to be rough, but they're not rough if you touch them. The appearance of being rough is because of organelles that we call ribosomes. That's where the proteins are synthesized in the body. Now, what does that have to do with Mensiferin? Now, let me show you back to this. Now, when you have the mitochondria under stress, everything else that are connected, you know, everything in the cell are connected, they become under stress, and vice versa. When the endoplasmic reticulum is stressed, the mitochondria is also stressful. It's almost like, you know, at home, if you're stressful, your spouse becomes stressful. Or your spouse is stressful and you become stressful. Sort of like that. So, taking another look at it from a different angle, if you look at the blood vessels, there are simple cells that are called endothelial cells in there. Okay? This is tied very directly to when you have diabetic or high blood sugar, those endothelial cells become dysfunctional. Okay? And sometimes they end up retard the flow of the blood circulations. You can see it ties to diabetic and also tied to hypertension. So if you take a look from a different angle, here you can see the cells are lined up. When one cell is stressed and a whole bunch of them have become stressful, almost the whole vessels are stressful. But there is a good tolerance that the body has, and it's useful to have a plant like Salacia reticulata with this kind of compounds in it. Now, here's what I was talking about before. Now, here is your rough endoplasmic reticulum, and then they produce a lot of oxidative protein. Okay, and then the rough endoplasmic reticulum becomes stressful. And when they're stressful, they release a lot of things into the bacteria, which make the bacteria become more stressful. If you look at it, they have excess of calcium beyond the normal level, the free radicals, and the cytochrome C. When you see a lot of cytochrome C in the blood circulation, that means one of the things that means is the mitochondria have been compromised. If you have a lot of the mitochondria that are compromised, that means low energy production, and it also affect a lot of other area in the body. That is very important. So in this research, they were looking at it, okay, if you put glucose into these cells, will you see the stress? Sure enough, when amount of glucose are excess, the endoplasmic reticulum becomes stressful, and it increases the production of reactive oxygen species or oxidants, which also increases the oxidative stress. And the oxidative stress level going high, it also increases the expression of a protein that we usually call DXNIP. Okay, and that protein actually turn on what we call a inflammasome activation. It's kind of like this. If you see here's a cartoon representation of what a inflammasome looks like, well, or supposed to look like. And when that thing is turned on, then it leads to inflammation. And for the purpose of discussion, all human diseases are results of inflammation that are not being controlled by the body. So there are certain type of inflammation, certain level that are useful, but beyond that, then the body suffers from the effects of inflammation. Okay, so and that leads to our discussion tonight: death of the mitochondria, or what we call apoptosis. Okay, and the mitochondria get rid of whatever that are inside and dies. Now, in this research, they add the Salacia reticulata and actually inhibit the stress, inhibit the reactive oxygen species, inhibit the oxidative stress, also inhibit the inflammasome activation. So this research is particularly useful because it gives us some idea of how it works. Okay. Now, another important factors that are involved in Salacia reticulata, looking at the molecular level, under A, this is how it works. Okay, you have the glucose outside, and you have insulin receptor, and you have what we call the GLUT4 enzymes, or called a transporting enzyme, or GLUT4 transporter. That's how it's supposed to work. When you, when the insulin is not there, then the glucose molecules stay outside the blood, okay, the blood vessels and cells. When it stays outside, it increases, of course. That means your blood sugar level goes up. When the insulin is released as a normal part of the process, then it activates the transporter. It's called GLUT4. Okay, so when it activates it, it becomes close to the ends, to the bilayer or the cell membrane, and it activates it. Now all the glucose are coming into the cell. And of course, a lot of them go directly to the mitochondria. The mitochondria burn them all out, release the energy that you feel. Okay, so that is very important. So Salacia reticulata also increases the activity of GLUT4 enzymes, which is another mechanism to help us understand how this plant is able to help us with the management of blood sugar. So I think we have about 10 minutes. We'll probably have to skip some of these slides. But looking at L-arginine, here are 4 ideas that I want you to get out of this slide. Okay, L-arginine are needed by the endothelial cells, the same cells that we were talking about before. And when the older we get, the less L-arginine are in there. Therefore, the blood vessels do not have the tone that we had once we were teenagers. Okay, so it's very important that you have that supply. So L-arginine helps with the vascular tone and also with the insulin secretion mechanisms. And then also, it helps with the heart health and your immune health. So this is a compound that is very useful. And again, the older we get, the less amount of that compound we have. And it contributes to a lot of diseases in both men and women. And particularly in men, is well known for the sexual dysfunctions. That is tied directly to the nitric oxide that contributed from this compound, L-arginine. Okay. The L-asparagine is very interesting because, of course, it was isolated from asparagus. But I want to point out 4 main health benefits from here, and it really helps with your liver function. Again, I think we have seen that with the Salacia studies before. It helps with your kidney function. It helps with the maintenance of the body and brain function. Okay? So it helps with the nervous system and all of those kind of things. Quercetin. Quercetin is present in a lot of plants and fruits. Again, we're going back to the this is one important aspect here of quercetin in the arteries. And if you look at it just from a compound microscope, you look at something like this. And when you have a lot of the oxidants and a lot of those things are hanging around, it actually contributes to, if you look at it from my right side here, here's how the healthy one's supposed to look like. And when its calcification process takes place, on my left, you can see a lot of white stuff going on around there. Now, you know that when its calcification process takes place, the blood vessels become calcified, and also it's hard to accommodate the flow of blood. When it's higher, then it should be able to open wider, and when it's low, then able to go down. So when you have that process happen, and you take a look at the examples, it actually problematic for the artery and the veins. Okay. Now, actually, there are two things happen. So the cursor shows the oxidative stress goes way up, and it leads to atherosclerosis. It also leads to what we call the process of mitochondria fission. So instead of growing, which is called mitochondria fusion, two mitochondria combine to become one, kind of like two powerhouses combined to make it more powerful. The process is called mitochondria fission. It breaks down one mitochondria to three little mitochondria. They are not very useful at all. Okay, so that's what you see with these kind of things. Now, when you add quercetin to the process, it actually inhibits the oxidative stress, inhibit the process, have been shown to inhibit the process of mitochondria fission. And then the smooth muscle cells, you have to see it in a microscope, actually assume their natural position and the color of the natural smooth muscle cells. That's an exciting research. So, how we do with that? Okay, let me skip that. Well, I guess I cannot skip that. There's an interesting study that we're looking at how the quercetin compound able to help mitochondria, and look at exercise tolerance. And then they have mice studies, mice that are running on the wheels and also treadmill. And then they look at they take the brain out and they look at it, and they also look at 4 different things that are associated with the mitochondria: the BGC1 alpha, the sirtuin-1 that are associated with the life extension and things like that, the mitochondrial DNA, and of course cytochrome C. A lot of cytochrome C in the blood circulation, meaning a lot of mitochondria have been damaged and are dead. Okay, so what they saw was something remarkable, and I wanted to show that. So quercetin, according to their study, were able to do the following. It increases significantly the BCG1 alpha, increases significantly the sirtuin-1 and mitochondrial DNA, and also the endurance capacity of these mice were actually increases significantly. Those that were in the doing the exercises, okay? Those who are running the wheel and those who are running the treadmill. So, coenzyme Q10 is an important compound that is, the older we get, the less the amount that we have compared to when we were teenagers. Coenzyme Q10 is very important for the cellular function, i.e. the mitochondria and health of the cells. So here are a few free examples of what they do. Number one, very strong anti-inflammation by it binds to the master switch in the cell, either will switch to antioxidant or switch to inflammation. So it binds it and turn off the switch. Number two, it prevents the, remember when the mitochondria is stressful, it opens itself and get rid of all its ingredients inside. So CoQ10 actually prevent that destabilization problem that happened in the bilayer of the mitochondria. And last but not the least, it also increases the energy, as it shows here. Here is the mitochondria in the middle, okay? So coenzyme Q10 is needed for energy. And this is another reason why the older we get, the less energy we have. The older we get, the less CoQ10 we have. Therefore, we need a little supply. But it also shows 4 things that are helpful. It helps with the liver functioning normally. It helps with your brain functioning normally. Helps with your kidney to function normally. And last but not the least, it helps with the normal functioning of the heart. Okay. The fenugreek seed extracts is an interesting seeds that are very important. And here's another picture of the mitochondria. And it also helps with that we have talked about before, and HbA1c. So the 3 things that come out of this study, it helps restore the activities of the mitochondria enzymes that are inside. It also helps lower the blood sugar level significantly, and it also decreases the HbA1c, which is tied to, that's another example of glycation. More sugar in the blood circulation. If you're diabetic, it means more glycation and products, which means more disease that comes. So people who died from diabetes are not necessarily died from diabetes itself. They die from other complications associated with diabetes, the heart, the liver, among other things. So in this study, what they found is the extract of the seed actually inhibit the LDL, the IDL, and also the triglyceride. Okay, they were looking only for those 3. So it's very important to help with the cholesterol level and significantly reduce those things. The cinnamon, I'm sure a lot of people are familiar with those. It's an interesting study. I said this study because they wanted to see how fast cinnamon extract can actually have an effect on somebody. So they were looking at this study. They pulled 7 male. It was randomized. It was crossover study. And then they have cinnamon and placebo. And they were looking at short-term effects. But they were surprised. It actually lowers the total plasma glucose level significantly. And it was immediate, and it was sustained for up to 12 hours, and also increases concomitantly the insulin sensitive level significantly, and it was also immediate and sustained in 12 hours. Okay, and here is your citation here for those who are curious to look at the study and read it. The alpha lipoic acid. This is an interesting study, and I just wanted to point out 2 things. Okay. They actually used diabetic type 1. 71 children and adolescents were of diabetic type 1, and it was a double-blind and randomized. And they wanted to take a look at the endothelial dysfunction because it's associated with diabetes, well known. Okay, so they have interesting things. They have an antioxidant diet by itself. They have a regular diet, and they have an antioxidant diet plus the alpha lipoic acid. And they found only the effects when the antioxidant diet is also together with the alpha lipoic acid, they found it improved the endothelial function significantly, and significantly reduces the bolus insulin reduction that you have. And I put here a little graph. So you can see when you have breakfast, you have a big spike of the insulin that are coming out, and then lunch, and then dinner. So those are the kind of things that contribute beyond the normal, the basal level. It helps, and it does contribute to the diabetic conditions. Okay, so it reduces that kind of spike from the insulin because it helps control that blood sugar level. Perhaps the enzyme inhibition, the alpha glucosidase, and also perhaps it activates the GLUT4 transportase enzymes. Okay. Okay. So basically, the anti-aging, anti-glycation of all of these ingredients, that are in the SRQ. It's very important to note them. So I'm sorry if it seems like I went over a couple of minutes. I apologize for the going over the time. Brett, please forgive me. But I wanted to share at least some of those things with you so that you understand that SRQ is a great product. By the way, I take that product at 2 in the morning and 2 in the evening after I eat my breakfast and dinner. I feel good. I love this product. I hope you love it too. Thank you, Brad.
Thank you, Alpha. That was a great presentation, and I'm sure everybody really appreciated it. It was kind of fun to see that all the attendees stayed right up until the last minute, and they're all still there now, hanging on every word, trying to understand and really understand this product and what's behind it. Obviously, there's amazing things behind the product, and we just appreciate your input and your explanation here tonight. We'll have this presentation available in the back office as quickly as we can, and I appreciate everybody being on the call tonight. And with that, I'll say good night and have a Merry Christmas if we don't see you between now and then.
Thank you.